So just as a this recap, I'll talk a little bit about what is electrophoresis and so on. So as we learned, electrophoresis means the migration of a charged particle in the presence of an electric field. So that means to carry out electrophoresis, whatever particle you want to analyze, it should have some charge, right? And within the biomolecules, this means we can analyze things like amino acids, peptides, proteins, nucleotides, nucleic acid, and things like that. And obviously, because it has to move in the electric field, how it migrates will depend upon the charge of the molecule. Some general terms related to electrophoresis. We will apply some voltage across the electrodes, which will generate some potential gradient. And the potential gradient is equal to the applied voltage divided by the distance between the electrodes, right? If we have any molecule with charge q in this gradient, it will experience a force, right? It will want to move towards the opposite charge, and will have the driving force F, which you can get by multiplying this potential gradient by the charge. Now it will also face some resistance, and this resistance will depend on the size and the shape or the pore size of the medium, so on, of the particles. So not all particles will move in the same way in the presence of this electric field. So we can define a term called electrophoretic mobility, which is basically how fast it can move in the field. And that is defined as the velocity of the molecule divided by the potential gradient. Okay. This electrophoretic mobility is what we'll refer to time and again in this course. Again, just quick recap. What we generally use in our lab is something called zone electrophoresis, which means that we have two electrodes. We have a positive electrode and negative electrode, and between that we have some supporting medium. It can be a gel, so it can be an agarose gel, it can be a polyacrylamide gel, it can be a piece of paper, it can be anything. But basically our sample is going to travel through this supporting medium, and because it is going to encounter different pore sizes, it is going to usually separate on the basis of its size. Okay. Again, there are two main types of gel electrophoresis. This is a horizontal gel electrophoresis, and this is a vertical gel electrophoresis. So electrodes are at the top and bottom, and the sample is traveling vertically. Okay. Again, how we run a vertical gel electrophoresis, so something like a polyacrylamide gel, which we run for proteins. We have to cast the polyacrylamide gel between two glass plates, and then we'll use this comb to make wells on the top where we can load our sample. Okay. And then when we have to run our sample, we will add whatever buffer we want to add at the anode, whatever buffer we want to add in the cathode. We have to load our sample in the wells, and then we'll apply a gradient so the sample can move through the gel. Okay. And we can separate our sample. Okay. Today I'll just talk briefly about what is the normal SDS that we use and what are the variations we can do in this SDS page procedure, right? But first we need to understand what we can achieve using our normal SDS protocol. So this is a very standard protocol called Lamley protocol, in which our sample buffer itself contains SDS, right? And what SDS is doing, so if this is our protein, it is going to be completely coated by SDS. So the protein is going to be completely linearized, and it's going to be covered by this negative charge. Okay. So all proteins now have the negative charge. Okay. So since now all proteins have the same charge, any separation is going to occur on the basis of its size as it moves through this gel. Okay. Now this running buffer, if you see the running buffer for this normal SDS system, we have the same buffer in both cathode and anode, which is tris, glycine, and SDS. Okay. And what happens is, you must be familiar with the term stacking gel. We load a sample here, and here, because we have glycine in the running buffer, glycine will have both positive and negative charge in the stacking gel. Okay, because stacking gel's pH is 6.8, which is close to the pi of glycine. So glycine has both positive and negative charge, or rather, there will be more of it will have a dipolar nature, and so it will move slowly through this region. Whereas the chloride will move fast. and all our proteins will get sandwiched between the glycine and the chloride. Okay, and this will force the proteins to form a stack. Okay, that is the purpose of the stacking gel. The proteins will all get compressed to a stack, so then they can be separated uniformly. Okay, so all the proteins will be focused here, right? So if you've ever seen a gel running, you will see all the bands travel together like this in the stacking gel until it comes to this boundary, right? And here we have this region of a higher pH, 8.8, and at this pH now glycine is mostly negatively charged, which means that it will also travel much faster than this. So there is no stacking effect in this part of the gel. It's now called a resolving gel. And because of that, proteins will now migrate freely and they will be able to separate based on their size. Okay? So this is just the theory behind a normal SDS pH gel. Okay? Now, when we load a sample in an SDS pH gel, we are basically completely denaturing the protein. Okay? Any subunits that we have will be broken apart, and we can even add additional reducing agents like beta-mercaptoethanol. And what this will do is, so if there are any disulfide bonds in our protein, what beta-ME will do is basically break this disulfide bond by reducing both of the cysteines. Okay, so in this way we can completely denature and separate the subunits of our protein, of any protein complex. So just to understand this a little bit more, we'll discuss this question here, where they say that there is some protein X. It has molecular mass of 400 kilodalton. Okay. But when we are running it on SDS page, it is giving 3 different bands. Okay, that means there must be at least 3 different components of this protein X. It has 3 different bands with molecular mass 180, 160 and 60 kilodalton. Okay. So we can draw what this will look like over here. So it has some band at 180, it has some band at 160, and some band at 60 kilodalton. Right? So there has to be at least 3 different subunits in this protein. Now again, they have run it on SDS page, but they have added the reducing agent beta-mercaptoethanol. Okay, and they are seeing some difference in the banding pattern. So they're saying when we run it with beta-ME, this time the molecular masses are 160, 90 and 60 kilodalton. Right? So that means that somehow this 180 kilodalton band has become this 90 kilodalton band. Right? When we have added beta-mercaptoethanol. So how can this happen? So the most logical explanation is there was some dimeric subunit of this protein which was being held together by a disulfide bond. Okay, both of these subunits have molecular weight 90 and in between there is some disulfide bond. But when we add beta-mercaptoethanol, this bond is being broken and we are getting both these subunits are separated. And because both of them are 90 kilodalton, they're coming in the same band over here. Right? So this means that this protein actually has 4 subunits. Okay? One is this 160 kilodalton, one is 60, it has 2 subunits of 90 kilodalton. Right? So this is option B. I think protein X has 4 subunits with masses 60, 90, 90 and 60. And if you do the addition, this will come to 400. Right, which was the original mass of the protein. Okay. If you have any questions, feel free to unmute yourself and ask, or you can drop them in the chat, either way. Now, here what we are doing is completely dissociating any protein complexes. But what if we want to keep the protein complex together? Right? What if we want to study if the protein is a dimer or trimer or something, what if we want to know that? So that we can do using another phenomenon called native page. Here we are studying the native properties of the protein. Okay, we are not going to denature the protein at all. So it's going to retain its original size, its original charge, and whatever subunits were associated in the protein, they will stay like that only. right? So we are not going to use SDS. We are not going to use any reducing agent, and we are not going to boil the sample. The sample is just going to be loaded directly in the gel. So in this, what is going to happen is the proteins are going to be separated on the basis of overall size, or on the basis of their size as well as their charge, right? Since we are not using SDS, we're not adding any charge to the protein. So the original charge is still going to be present. Now, because we are not changing the protein, it can still retain its function. So we can also use these for enzymatic assays. Okay, those are called zymograms. So just to help you understand that the charge of the protein is also retained, let's talk through this problem. We have a native gel in which we are going to have proteins A, B, C, and D. Okay. Now A, B, C, and D have equal mass and shape, let's assume. But they have different isoelectric point. Okay. They're running them in a buffer which has pH 6.4. So now we have to understand what charge will these proteins have at this pH and how that will affect its migration. Okay. So now pH 6.4. What are the PIs of our proteins? 5, 6.4, 7, 9.2. Okay. So now if pH is less than PI, is less than the PI, the protein will carry a positive charge, right? If pH is greater than PI, the protein will carry a negative charge. And if pH is equal to PI, the overall charge on the protein will be 0. Okay. So considering this, let's try and see what will happen to A, B, C, and D. Now PI of protein A is 5.2, which is less than 6.4. Right? That means that protein A will probably be positively charged in this situation. So A will have to migrate towards the negative pole, right? Protein B has pH 6.4, but the buffer also has pH 6.4. So that means it is at its isoelectric point only. So there is no charge on protein B. Now think about the definition of electrophoresis. It is the movement of a charged particle. So if we have any uncharged particle, it will not move at all. So that means B will not migrate in this scenario. Protein C and protein D have PI which is greater than pH. Okay, which means that they will be negatively charged. Okay. So this is positive, this is 0, this is negative, this is negative. So protein A is positively charged, so it should migrate to the negative pole. Protein B will not migrate. C and D are negatively charged, so they will migrate towards the positive pole. Okay. So here option D is the correct option. Okay. I hope this is clear to everyone. They're just showing that if we have a native gel, then proteins will also migrate based on their native charge. Okay. Now what if we only want to study the molecular properties of this protein, right? Here we can see that the charge is also having an effect on the migration, right? But what if I only want to study what is the subunit composition? Is it a dimer, is it a trimer, etc.? Is there some other way I can have a uniform charge on the protein? Yes. Okay. This is something called BN page. Okay. It is called blue native page. Where we are basically using a different molecule to impart a negative charge to the protein. Okay, this is a dye called Serva Blue G. We can also use Kumasi R250. And when we add it in a sample loading buffer, it basically surrounds the native state of the protein and gives it a negative charge. Okay, but it is not going to denature or dissociate the protein. Okay, we are just giving uniform charge to the protein. This means that the protein is now only going to migrate based on its mass. Okay. One thing about this native page is that we have to use different cathode and anode buffer because we need to cause a stacking effect to occur somehow. So here in this scenario, we use histidine as the trailing buffer. Remember, we had used glycine earlier. Use histidine as the trailing ion, chloride as the leading ion, and in the same way, the proteins can be separated based only on their size. Okay, so large proteins will be stuck here, smaller proteins will be down here. Okay, one other thing we can use to separate these proteins very well is to cast something called a gradient gel. Now, you might be familiar with, say, casting a 10% gel or a 12% gel, where the whole gel, the acrylamide percentage and the whole gel is equal throughout. Right? You're just making one solution, you're pouring it into the gel cassette, and it has the same percentage throughout. But if you want to separate a large range of molecules, what we can actually do is cause a gradient gel. Okay? Which means it has a low percentage here and a high percentage here. So that way many molecules of many different sizes can be separated. Okay? And just an example of what this kind of gel can look like. Okay? Now we can run BSA, which is a monomer itself and has molecular weight of around 70 kilodalton. So you can see that here. But consider another protein called GAPDH. Okay, it has molecular weight of 36. So if I run this on a normal SDS page gel, I will get a band at 36 kilodalton. Right? But when I've run GAPDH on this gel, I am not getting any band here. Right? There's no band at 36 kilodalton. Instead, I'm getting the band all the way up here at 143. Why? Because GAPDH is a tetramer. So its total molecular weight will be 36 into 4, which is 143 kilodaltons. It's why I'm getting this band here. Okay? So just an example of what a native page gel can look like. We can go through this example of comparing a native and SDS page gel to see if what is the nature of our protein. Okay. So sample 1, 2, 3, 4. Again, we have run the same sample 1, 2, 3, 4 on a native page and SDS page. Right? So remember, in SDS page it would be completely denatured. In the native page, the original subunit association will be retained. Okay. So SDS, let us consider sample 1. SDS page, we're getting some band around, let's say 60. No, it's close to 63. We'll call it 60 kilodalton. Here, we've got the band at 180, which suggests that 16 to 3 is 180, right? So how will I find how many subunits are there? So number of subunits will be equal to weight in native page upon the weight
Homotrimeric.
Yes, correct. SDS. So this will be a trimer, right? I'll just write 3 for ease of understanding. It's a homotrimeric protein. Let's consider sample 2. Okay. Here I'm getting some band at 75 in the SDS page. Even in the native page, I'm getting a band at 75, which suggests that this is the single subunit protein of 75 kilodalton. Okay. Because I'm not getting any change in the weight and I'm not getting any additional bands also. So this must be a monomer. If you look at sample 3, this I'm getting in SDS I'm getting a band of, let's say, 30. Here I'm getting a band of, say, 60. So 60 by 30 is 2. So this is a homodimer. Okay. Same way, considering sample 4, let's say this is 20 and this is 80. 80 by 20 is 4. So this is a tetramer. Okay, so this is a very easy way to kind of get an idea of how many subunits are there in any given protein. Just run it on an SGS page and a native page. Okay, I thought that uh, uh, yeah, while we're here, I can show you how people run a native page. I can't do it in my lab because a lot of people there, but yeah, I thought I can.
Avoid turning the tube right side up. I fit into a new chilled microcentrifuge tube and leave it on ice until loading of the gel.
Wait.
Gradient gel pouring.
Wait, what happened to this?
I mentioned denaturing SDS tube. Close the cap and cut
Right. Okay, fine.
off any excess dialysis membrane that's
sorry about this. I don't know what has happened to this video. But this is actually a very nice journal called the Journal of Visualized Experiments, okay, called JOVE, where they usually have a video for any kind of experiment you can think of. So they will demonstrate the video for you. I'll just reload it one more time to see if it will play.
the overall goal of the following. As a second step, the lysate is applied to first dimension. For second
Apparently not.
dimension it.
Okay then. Interesting. Okay. Instead, maybe what I'll show you is how to cast a gradient gel.
Hi, my name is Celeste
So,
Peterson.
if you remember, I discussed earlier that we all know how to cast a regular gel where the percentage is the same throughout. But what if you want to cast a gradient gel? So here I think they've discussed how to cast a gel with an acrylamide gradient. Okay. So first, this is some preparatory step that we don't need to worry about. Okay, this is very specific for her experiment. Okay, and here she's going to describe how to cast a gradient gel.
Feeling around for impurities and trying to fill mud so that syrup. Anything like that can cause irregularities in the gel. It's important to wipe off all the water. So when I assemble the gel, I will put that chip on the outside of the gel. So the gel will be in between these two pieces of glass, and the chip will be on the outside, and it won't affect the gel in between. So now I'm going to assemble the gel in these clamps. And then I squeeze them together and tighten by turning to the right. As with any gel, it's important to make sure that the bottom is flush to prevent leaking. We now turn the gel around and put it in here.
No, we can't hear the video.
You can't hear the video, is it? Okay, fine. I will do. You can hear my voice, right?
Yes, ma'am.
Okay, then I'll do live commentary then. So
Get sealed with a
she's
gasket. So it's
just
important when you do
assembling the
this,
gasket. So
that the
this is the same for
is lined up with the gasket here.
any gel basically that you want to run. But you'll see that she's cast a very big gel,
These
right?
knobs
So
on the side,
you can
then
the
go
size of the
in
gel that you take will depend on the nature of the proteins you want to separate. But obviously, if you want to separate things with very high resolution, it's better to cast a big gel.
So this is a 30 ml syringe.
So here, what she's actually going to do is prepare two solutions. Okay, one with a high acrylamide percentage and one with a low acrylamide percentage. Okay, so and she's going to fill it in these syringes. So yeah, here she's just washing the syringe.
To put the tubing
Okay,
onto the gel, to load the gel, first you put an adapter into the tubing.
and now she's just connecting the tubes to a needle,
Small adapter.
and the needle is going to go in between the gel plates, and that will deliver the gel solution.
Just so it's barely below the bottom glass.
Okay,
Then this Y adapter.
and now if you see that plastic wheel in the back,
And
that is what she's going to use to make the gradient.
syringes containing the high and the low density solution. So these are
Okay,
pre-prepared
so
solutions.
in the beaker there are those two different solutions with two different acrylamide percentages. She's put blue dye in one of them just to show that that is the higher molecular weight one,
This is the least in nature.
and that is the lower acrylamide percentage one.
And they have
So she's just kept and prepared the gel sample. Now she's going to add TMED. Right? Remember, TMED is required to polymerize the gel. So now only after she adds TMED, the polymerization process will begin. And now she will have maybe about 10 minutes to prepare the full gradient.
And once you've added the 10, you have about 5 to 7 minutes to fill the gel before.
Just mixing it well.
Now we fill the syringe.
Now she has to fill the syringe.
So I'm going to fill it with about 15 ml of solution. And then to get rid of any air bubbles.
And obviously we don't want any air bubbles in the middle of our gel, so we have to remove the air bubble.
Air bubble is gone. Now this little screw here is going to go into this groove. This adapter is going to line up
Okay, so
with
she's just attaching the syringe to this sort of gradient mixer.
very tightly. And then this tubing is going to attach to the syringe.
Okay, and same thing is going to happen
Other side
with the
high density.
high density solution. Okay, same thing, we have to remove the air bubble. Okay, now both solutions are prepared, and now we can start making the gradient.
Where it's time to deliver. And so I'm going to be turning this wheel in a steady clockwise motion.
So as she starts running the wheel, first you'll see that only the blue solution is going in first, right?
And then after a little while,
So that means that the highest percentage of acrylamide will be at the bottom.
1 moment.
And over time it will start delivering part of the lower percentage. So as it mixes, it will form a gradient up the gel. Okay, you can see the color gradient quite easily, right? So this is how we can cast a gradient acrylamide gel. Okay, so this is better if you want to resolve compounds over a very large range of molecular weights. Okay, I think someone wanted to. Okay. Where were we? So you can imagine that if I have complexes which are very big, like 500 kilodaltons, if I run it on this gel, it will get stuck somewhere here. I won't be able to resolve it at all. So in that kind of scenario, it is better to cast a gradient gel. Okay. So sir also this week talked about something called 2D gel electrophoresis. And if any of you were around for the last live interaction, we had discussed a little bit of that, but we can go into more detail today. Okay. So 2D gel electrophoresis starts with a process called isoelectric focusing. And what that means is separating proteins based on their isoelectric point. Okay. Remember, isoelectric point, or pi, is the pH at which the protein has overall zero charge. Okay. So what we have to do for isoelectric focusing is first form this pH gradient. Okay. So we have a strip with a pH gradient. It has low pH to high pH. So let us assume pH 4 to pH 9. And when we have to add a sample on this, and when we apply a current, what is going to happen is that these will move on the basis of their charge, okay, on the same way we had discussed for that native gel earlier, right? These proteins will move on the basis of their charge until they reach their pi. Okay, so here we can consider this yellow protein. Okay, when it is on the low pH side, it is positively charged, so it will move towards the negative electrode. And when it is over here, it is negatively charged, so it will move in this direction until it reaches a point, or it reaches a pH where it has overall charge 0, right? And now there is no longer any electromotive force acting on this protein, so it will stay over here, right? So it will stay at this pH which is equal to its pi. So in this way we can separate the protein mixture. Okay, this is just a process called isoelectric focusing. The pH gradient is generally made by these things called ampholites. Okay, so we will add a mixture of a sample and an amphylite on the pH strip, and they will separate to form the pH gradient. Okay, this is basically the same thing. We have a pH gradient, we add a protein solution, apply electric field, and the protein solution separates into its constituents based on their pi. So this is what we call the first dimension in 2D gel electrophoresis. What we can do is then take this isoelectric focused gel, okay, and just add it to a normal SDS gel, right? So now we already have all these separated proteins. What we can then do is run it in a normal SDS gel and separate it based on their molecular size, right? Sorry. So in this way, the first dimension is separating based on pi, second dimension is separating based on size. So in this way we get this very complex looking gel where you can separate here more than 1,000 different proteins. Okay, so here, as you can imagine, everything in this band, okay, has the same isoelectric point, but they may not all have the same mass. So if I look at all the proteins which are there on this line, okay, these are all proteins with the same pi, but they have different mass. In the same way, if I look at all the proteins here in the same horizontal axis, right, these are all proteins with the same mass, but they have different isoelectric point. Okay, and by this method of two-dimensional electrophoresis, we have separated based on two different criteria, so we are able to get a much better separation of these gels. And so what, I mean, if you're thinking what is the application of this, right, it looks very complex, what can I possibly do with this? What you can do is, if you're interested in any specific one protein point, say I want to know what this protein is here, so I have now separated it very well from its other proteins. I can actually cut out this protein, this piece of gel, and use it to sequence this protein and find out what it is. If, say, I have two different conditions, okay, I have some plant and then I treated the plant with some drug and I want to see if any effect is there, then I can run a 2D gel for both of these conditions and see if there's any overall difference in the protein expression. Okay, so the 2D gel electrophoresis can be a very useful tool depending on what your application is. Okay, I wanted to show you a video for 2D gel electrophoresis. We'll see if okay, I'll see if you can hear the just let me know if you can hear the audio, otherwise I'll do live commentary again for this. Okay, this is just showing us how to run a 2D gel. Okay.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good, reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip.
It's not audible, no?
The second segment will be on how to equilibrate the IPG
laptop's
strips in
mic, then
preparation
we
for
will
running on
able
the second
to
dimension
hear.
gel.
Yeah, so the laptop's mic is unmuted, so yeah, I'm not sure what's going on. Wait. Da-da-da. Input by default. It's okay. So I'll talk over what he's explaining. It's partly a commercial for Biorad, so you won't get to hear him promote Biorad products, but I think that's okay.
And the third segment will be.
So here he's just giving an introduction of what
The first step
the gel looks like.
to rehydrate the IPG
So
strip. And I'm going to demonstrate this
first he's going to show us how to use the strip that we use for focusing.
2D gel electrophoresis.
Okay, so for isoelectric focusing we use something called an IPG strip. So that already has a pH gradient established in the IPG strip, and he's going to show us how to prepare the strip for our experiment.
Chaps, DTT, ampholyte, and
So here he's showing a resuspension solution which contains the ampholytes. Okay, so we have to first mix our sample with the ampholyte mixture, which is called the rehydration buffer.
To rehydrate the strip, I will load 200 microliters of sample into the focusing tray.
He's saying for this particular strip, this is 11 centimeter strip, we need about 200 microliters of sample. This plastic tray you can see is called a rehydration tray. You can see all these different slots, which is basically different slots for different samples. So we will just add our sample into this tray. Okay, and then just soak the strip in this sample.
Along one edge of the lane of the tray.
Right, he's just loading the sample.
In a nice even bead from one electrode
right, you have to just
across to the other.
Fill this tray with the sample evenly, so that it can be evenly distributed on the isoelectric focusing strip.
Tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG
this is what the strip will look like. Just a
An
clear
IPG
plastic
strip
gel.
has 2 sides. One is a plastic protective
There is a
backing,
plastic
and the other
protective
is a dried
piece
gel.
that we have to pull off,
You begin by
and the
removing
other side
plastic
is the gel.
protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces.
So you're just going to
that has indented is your protective backing.
just going to pull it off.
You pull that rapidly across, and now you have gel side up.
So this is the side with the gel.
Take your forceps, hold onto the strip on the negative side.
And
You'll see the other side has a positive side. The negative side
on
is
the strip,
opposite
it's
of
already
that.
marked, which is the negative and
you can
positive
always
side.
tell when the IPG strip is facing down, you'll be able to read the numbers.
And it
of
is
the
also
pH
marked with
range
what
of
is the
IPG
pH
strip
range of the strip.
in
So
the
this is
correct
pH 4 to 7.
orientation. Now the gel will be loaded onto the sample bead gel side down.
Okay,
If you're
and
running multiple samples,
then we have to
I
now
recommend
basically just
labeling your strip
load this
before
onto
the
our
strip.
sample. So it
Make
will
sure
just
to
be
label the
kept
side of
on the
gel
sample.
and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray,
Okay.
and slowly drop
So
the
just
IPG
slowly putting
strip
it
into the lane of the focusing
into
tray.
the lane. you have to make sure that you line up the positive and negative side. So if you see these dark lines, those are actually the electrodes. So this tray has a positive and negative side. We have to match that with our strip.
If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your
Okay,
first
and
dimension
it's just saying you have to
isoelectric
remove air
focusing
bubbles
run.
properly, because
The
otherwise the
initial
sample will
step
not
of
be
the first
applied
dimension
onto the
separation is
strip,
to rehydrate the
and so
strip.
it won't
I
separate
will
properly.
do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight
Okay.
proteins.
And here we're just soaking it in our
sample,
sample, so it's called passive rehydration. But if you want, we can apply a small voltage which will force the sample to go into the gel, which is called active rehydration. But that's not necessary for most samples. So now we are going to actually perform isoelectric focusing. Here on top he's putting mineral oil, which is to prevent evaporation. Okay, it's to make sure that the strip does not get dehydrated during this process.
Out during the course of the rehydration and the run. Now I am going to program the protein IEF cell.
So this is the first dimension, which is isoelectric focusing. So charge is going to be applied to both ends of the strip.
Typical rehydration times are 12 hours, and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first, remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C.
Yeah. So we will just apply constant voltage on that for, as you said, around 12 hours or overnight, so that we can properly focus our proteins in the isoelectric focusing.
The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip.
Okay.
Each solution contains SDS for denaturation of proteins
Now
in the IPG
our proteins
strip
are properly.
to ensure optimal
Now,
performance in
if
the
you think
second
about it,
dimension.
the IF has to be done under native conditions, right? Because we want the proteins to have the native charge to separate it based on PI. But in SDS, it needs to be done in denaturing conditions, right? Because we want to separate based on molecular weight. So we have to do something called equilibration, where we have soak the gel in a buffer, which will cause the proteins to denature. So actually there are two equilibration buffers. So
onto the IPG strip.
Here he has removed the IPG strip. He's just removing any excess mineral oil.
Be careful not to press too hard.
Okay, mineral oil or excess buffer or anything. He's just removing it from the strip.
The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper.
Now he's going to do the equilibration, which, as I mentioned, is to prepare the strip for the second dimension.
Into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1.
So this is the first equilibration buffer. It contains DTT, which is going to reduce the proteins. Okay, it's going to reduce.
Place the rehydration tray onto a shaker rocker table for 15
now,
minutes.
It is going to be.
Note that the
we need to.
Rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray.
So it's now going to be kept on a rocker for about 1 hour.
Now add 2 milliliters of buffer number 2.
And this is a second equilibration
Shake
buffer.
for 15 minutes and pour out.
Okay, same deal. So this has iodoacetamide and SDS.
Run on the second dimension gel.
And again, we're going to incubate it for maybe 15 minutes, and then it is ready to run in a second dimension gel. So if you can see that SDS gel at the back, that is what we're going to use for a second dimension.
From the bottom of the gel,
This is a fancy lab in the US, so they have a precast gel.
And slide out the comb
But
from the
if
top.
we were doing it, we'd have to do it ourselves. You'll
Rinse the wells
see there's one very
with
big well,
good quality
right?
DI water.
Normally we would have many
Tap
small
the water
wells,
out
but here there's
into
one
a
big well
beaker.
where we can directly insert our strip.
And then place the gel sideways and remove with a piece of filter paper the excess water.
So here he is just
from
washing the well,
wells.
and
Be careful
he's
not
using
to
a piece of
touch
blotting paper to make sure there's no
bottom of the
water that is
gel, as
left.
it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed.
So it's just there to remove any unpolymerized acrylamide which might be left in the gel.
To remove the strip from the tray, place the forceps on the tray and
Now,
gently push
our
on the plastic backing
IPG
of the
strip is
gel
completely
to push
equilibrated,
the strip towards
which
the
means it's
angled
ready to
edge.
run in the second dimension.
Lift up the strip from the right tab.
So he's taken it out of the
I'm
tray.
going to rinse the strip off in running buffer that I have
He's just going to
prepared.
dip it in the SGS running buffer.
Dip 2 or 3 times. This will remove equilibration buffer number
that is
1
removing
and 2
whatever
that may
equilibration
still be
buffer
on
was
the
there.
surface of the IPG strip. Place the IPG strip.
And now we have to put
gel
the IPG
side
strip
up.
into that well that is
Place
there in the
plastic
SDS gel.
backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps.
We're just making sure that it's
And gently
pressed down
push on the strip
against the surface of the
until
well.
it touches the gel.
Right? There
If
shouldn't
you
be a
feel that
gap
it's
there
touching,
because it won't be able to migrate.
A little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose,
So
into the reference
now
well on the right
we
side
also,
of the gel.
when we run any SDS gel, we
Break
always run a
off
marker,
one
right?
plug.
That we get some idea of the approximate size of our protein.
Hold
But
on
here
to
there
the
is no
handle
well for us
and
to
break
load this
off
marker.
the spatula.
So what
And
he's
using the
actually going to
spatula,
use is
push out the
a
agarose
small
plug.
piece of agarose gel which already has the marker embedded in it.
For criterion
Okay, that
gels,
blue
I
thing
usually
is a
cut
piece
the
of
plug
agarose gel
into
with the
4
marker
slices
already
to
contained
match
in
the
it.
size of the well.
So we're just going
In
to
this
cut a
case,
small
I need
piece
1.
and add it into the well.
With forceps,
So this will
hit
act as the
one of
marker.
slices, place on the back of the
So
gel
same thing.
cassette over
There
the
is a
reference
small
well,
place for him to
and gently push
put the marker.
the
You're just going to
plug
push
into the reference
that
well.
reference
At
marker
this point,
into
we
that.
want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer
And now
hot
that
to the
everything
touch,
is pushed into
take
this
the hot
2D gel,
agarose
he's just going to seal
and
the gel from on
fill
top
up
something
the
with
well.
agarose.
Do
Okay,
this
this is called
one
overlaying with
rapid
agarose.
action.
This is just to make sure that it stays there
Side to
properly and
side.
doesn't move.
Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles.
Right? Just getting rid of any air
Agarose
bubbles.
is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some
Again,
of
if
the
you look at the
larger
top of this gel, it says 8 to
ones, and you should not have any
16%
problems for your second dimension
tris
gel.
HCl. Right? So this is
Now
again a
that the
gradient
agarose
gel.
is solidified, I'm
It's a
ready to
gradient
run the
acrylamide gel.
In this case, I'm going to run the gel in the criterion gel box.
Okay, now
which
he's just going
holds
to run
2
the gel.
gels. In most cases, you're going to run multiple gels, and you want to use a criterion dodecacell.
This I think is a procedure everyone is familiar with. It's just going to put the cassette into the running tank.
cell.
Running tank is filled with running buffer.
Load the upper buffer chamber with
It's going
running
to
buffer.
filled up the buffer chamber.
Place the lid onto the
Going to
criterion cell.
add the lid so that connects the
Insert
electrodes,
leads
connect it to
into
the
your
power pack,
power supply.
and
Program for 200 volts for 1 hour.
we're going to run it at 200 volts.
Constant voltage. And start. L has now completed running. You know it is finished when the dye front has reached the bottom of the gel. We're
Okay,
now
so
going
now
to
finished
remove the
running,
gel from the
and
criterion
now he's going
cassette
to
and prepare
stain the gel.
for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by
Okay, so he's just going
pushing
to
the forceps against the plastic cassette
remove the gel from the cassette.
towards you at the top.
Okay, so this is just
and work your way down. So fold open, like so. After I opened the gel cassette, I used forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the
So
gel
now we have to
upside down over the surface
take
of
the gel out of the caster.
staining solution.
So just using the
remove a
forceps to
corner,
do this.
And remove a corner until the gel falls into the solution.
Right, so now the gel has fallen out of the
Biorad
cassette into the
offers
staining solution.
many different staining
Okay, there are many ways you can stain the gel.
gels,
So discussed this last week in the course. So mostly we use Kumasi.
To the fluorescent stains,
If we want
flamingo,
to
and
have
cypro.
more sensitive staining, we
In
can use
this
silver
case, I
staining,
am
which is about
staining this
100
gel
times
with
more sensitive than
Biosafe
Kumasi.
and the
Or
first
you can
step
use
requires
fluorescent
3
dyes
10-minute
like flamingo and
rinses in
cypro
water
and things
to
like that,
remove
which
SES
are
from the gel.
extremely sensitive.
We are now going to stain the gel using Biorad's Biosafe Kumasi stain.
Okay, he's going to use
Following
Kumasi staining,
10-minute
right,
water
which is
washes,
just
we're
going to
transfer the gel to a tray of Biosafe Kumasi.
just
And we
transferring
are
the
gonna
gel to the stain.
gel for 45
Right,
minutes
that blue solution is
to
the
1
stain.
hour.
He'll stain it for about 1 hour,
Following staining, we are going
and
to
then we're going to
transfer the
destain
gel
it.
from Biosafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging.
Right, so we have to destain it until
Here
we
you
get
can see that I
rid of the
have
background, and
generated
here you
a
can see what the
gel
2D gel
with
looks
a
like.
clear background and well-resolved spots.
Okay, so extreme right. Yeah, we'll wait for him to come to the conclusion.
I have now completed the entire process for running a electrofreeze.
Okay, he's not going to show us the gel, so we'll go back. So here, extreme left, this was the marker, if you remember, that we loaded. And these are all the spots that we got in the 2D gel, right? Which looks familiar. Okay, so that was just, because it's not something I can show you, just an example of how we can run a 2D gel. And
I have 2 questions.
yeah, go ahead.
Ma'am, while he was doing the first dimension, like the IS,
Mhm.
so he used mineral oil. So
Yeah.
what was the process?
Right. So as you saw, we have to keep IAF for a very long time. Right? We're keeping it 20 hours, like 16 hours, 20 hours, and so on. So if we just leave it open, whatever buffer was there in that well will actually evaporate, and our gel will dry out. So because of that, we keep mineral oil on top, which is stopping the evaporation of the buffer. So just so that the gel does not get dehydrated.
Okay. And ma'am, the SGS stage, I mean the into 2D dimension when he was doing, the gel ran on 200 volts.
Yeah.
So we learned last class that the limit of the volts is 140. To be 140, otherwise heat will generate. So what about that?
Yeah. So, I mean, when I was talking last class, I was talking about 140 in relation to setups that we usually use in our labs here, right? And I mentioned there's also a process of trial and error. And I agree, if I was running the gel, I probably would not go above 140 or 150. But apparently for their setup, it is okay to run at 200 volts. So again, this is a trial and error based on the setup that you have. If I was running the same gel in my lab, I would not run at 200.
Okay, ma'am.
Yeah.
So does this come with the protocol or something like that?
So there will any protocol you get will have some value written, but that does not mean it will work in your lab.
No, I mean device protocol or something like that.
Device protocol, sure, yeah. If you get your power pack, it will come with some protocol for like how much heat will be generated and so on. So once you have your own device, you can
Okay. Okay, ma'am.
Yeah.
Thank you, ma'am.
No worries. So anyway, since we just learned about IF, we can try and solve some examples on this. Okay. So here is asking, at some point during isoelectric focusing, the protein stopped moving through the gel because dash. Right? So why is the protein stopping at that one point? Why is it not just moving through the gel and getting collected at the electrode? And that is because at that point the pH is equal to the isoelectric point of the gel, which means the net charge on the protein is 0. So there's no longer any electromotive force on that protein. Right? So the correct answer here is B. They have net charge of 0. Okay, net charge of 0 does not mean that there are no ionized groups. It just means that the number of positive and negative charges is equal. Okay, so the net charge is 0. Right? It has nothing to do with their mass. Okay, IPG strip is, we sometimes even run in an aggro strip. Okay, so it has nothing to do with the protein mass. It just has to do with the fact that the net charge of the protein is 0. Okay. So here the question is, we have this tripeptide, glutamate, methionine, aspartate, and it is asking, if we run it in an isoelectric focusing gel, where will it run? Right? So where will it stop? Obviously it will stop when the pH is equal to the pi. So here we have this pH gradient from 3 to 9. And so the question is, what is the pi of this peptide? So first let us think about which groups in this peptide can get ionized. Okay, any peptide will have a C terminus and an N terminus, right? So the C terminus is ionizable, it has pKa of 3.5. N terminus is also ionizable, it has pKa of 8, right? So basically it is saying that below pH 3.5 this will be COOH, and once the pH is above 3.5 it will go to CaO minus. And same way, below pH 8 this will be NH3 plus, and above pH 8 it will lose this hydrogen, so it will just be NH2. It will not be charged. Now you must have also learned that some amino acids have ionizable side groups. Right? So both glutamate and aspartate have ionizable side groups. They actually have very similar side groups, so it is this acid which will be ionized. So aspartate has pKa of 3.9, glutamate has pKa of 4.1. So the question is, how will we know what is the pi of this peptide? Okay, let us consider what will be the charge on this peptide at different pH. Okay, so let us just say, what are the pKs? 3.5, 3.9, 4.1, 8.0. Okay, these are the 4 different pKs in our peptide. So if I consider the pH to be 3, what will be the charge on this protein? What will be the charge on this peptide? Right? This C terminus is still not ionized, so this will be 0, this will be 0, this will be 0, but this will be plus 1. Right? This will be NH3 plus. So at pH 3 it will have charge plus 1. Okay. What if I go to some pH over here? Say I go to pH 3.8. Okay. 3.8 is above the first pKa, which means that this H is going to be lost and this will become COO minus. But this will not be ionized, this will not be ionized, this will still be NH3 plus. So what is going to be the net charge on this protein? This is going to contribute minus 1, this is going to contribute plus 1. So net charge on this protein will be approximately 0 at this pH, which is between these two pKa's. Okay, if I go to pH 4.0, let's assume, even this is going to get ionized. So I'm going to get additional -1 charge here. So I'm going to -1, plus 1. So final charge is going to be -1. Okay. Similarly, if I move beyond this pKa, even this... Okay, sure. I'll repeat from here. Okay. So this peptide, glutamate, methionine, aspartate. Okay, we have glue, met, asp. Okay. Okay, so this peptide, we want to know what is the pi, what is the isoelectric point of this peptide. Now, any peptide will have an N terminus and a C terminus. Okay, so this will be NH3 plus, and this will be COO minus, right? So this is how you'll be familiar with it at pH 7. So whenever we draw the structure of an amino acid, you must have seen this written as NH3 plus and COO minus. But that does not mean that this is the charge they will have at all pH. So what this table is telling us is the pH at which this hydrogen will be, this group will be ionized, right? So what this is telling us is that this will actually exist as COOH. And once the pH goes above 3.5, we will lose this hydrogen and it will become COO minus. Okay. And same way, this NH3 plus will be NH3 plus only until pH 8. And above pH 8, it will again lose one hydrogen, it will become NH2. And it will lose this positive charge also. Okay. So as we increase the pH, this is going to acquire the negative charge and this is going to lose a positive charge. Okay. Now, glutamate and aspartate, in this structure itself, they have this acidic group. Okay, which can also lose this hydrogen at different pH. So what this is telling us is at pH 3.9, this aspartate group will lose the hydrogen. So it is COOH, right? It will become COO minus. This has charge 0. This has charge -1. Okay. Same thing for this glutamate. Exactly the same. It's just that for glutamate, this process will occur at pH 4.1. So now let us find the pH at which this overall peptide will have charge 0. Right? That doesn't... So for that, let us consider the scenario at different pH values. If I start at pH 3, this is below all of the pKa's in our protein, right? Which means that everything is going to be in this state only. So the only charge which is going to be there in the protein is going to come from this amino group at the N terminus. So the charge of the overall protein will be +1. But if we go to a pH which is intermediate here, so let us say pH 3.8. Okay, what is going to happen at pH 3.8 is this, we are going to start losing this hydrogen, right? This hydrogen on the C terminus is going to we're going to start losing, and that means this is going to acquire a negative charge here. But the amino terminus already has one positive charge. So the pi of at this intermediate pH will be around 0, right? Because we're going to have +1, is going to be +1. This is going to be -1. So approximately 0 is going to be the overall charge. Again, if we go to some pH after the second pK, let us say 4, right? Because 4 is more than 3.9. That means even this aspartate side group will start losing its hydrogen, and even this will acquire a -1 charge. So now the overall charge would be -1, which will be -2, +1. Right? So overall charge will be -1 only over here. If we go to another intermediate pH, say pH 6, now even this glutamate has gotten the chance to ionize. So even this will become -1. So now the overall charge will be -3 + 1, which will be -2. Okay. And if I go to the extreme pH beyond 8.0, what is going to happen is this hydrogen from NH3 plus is going to be lost. It's going to become NH2. So there is no positive charge here anymore. So the overall charge will be on this will be just these 3 negative charges. So it will be -3. Okay. So now we have gotten an idea of what will be the pH of this, what will be the charge on this peptide at different pH. Now what we wanted to find, what is the isoelectric point? The point where charge is 0. So the exact isoelectric point of this protein is equal to the average of these two pKa's, right? It has to be between these two, obviously. So the exact pi here is equal to 3.5 plus 3.9 divided by 2. Right? We want to find what is the midpoint between these two, because here it is +1, right? Below 3.5 it is +1. Above 3.9 it is -1. So you want to know what is the midpoint between these two where it will have charge 0. So we will take the average of these two pKs, and so we will get the pi of this protein is 3.7. Okay. So now we know at pH 3.7 there will be no net charge, and that is where we will find this peptide in this isoelectric focusing gel. Okay. This already has pH gradient from 3 to 9. This is a pi of 3.7, so it will get stuck somewhere here. Okay, so we will expect to find it here. Okay, is this example clear now to everyone? Again, if you have any questions, feel free to ask me or put it in the chat or anything. Yeah, good. Okay, so now we got an idea of how the isoelectric focusing works. We'll try to answer some more questions. Here is a 2D gel. Okay, they've already run it and they're asking us to answer a few questions. So there are some 5 proteins, A, B, C, D, and E. And they're asking which proteins have highest pi value. Okay, good. Glad to hear. Yeah. They're asking which is the highest pi value. Right? So this gradient is going from 5 to 9. So it is asking which protein has the which protein is coming furthest to the right in this isoelectric focusing. So obviously the one which is coming in this column, which is protein D and E, will have the highest pi value. They are having pi of around 9. Whereas B and C are having pi of around 7 and A has pi of 5. So here the answer will obviously be proteins D and E have the highest pi value. Next is asking which have the highest molecular weight. They're asking based on the second dimension which protein is having the highest molecular weight. So we know in SDS page the proteins which have lower molecular weight will travel towards the bottom and the high molecular weight will be at the top. So whichever band is at the top here will have the highest molecular weight. So that is protein B. So protein B has the highest molecular weight. Now they're asking which proteins have identical molecular weight. That means that they are coming in the same horizontal line in this SDS page gel. And here the only proteins in the same horizontal line are A and D. Yeah, correct. So A and D will have identical molecular weights. Okay. And just one last example. Now we have an idea of what the 2D gel electrophoresis works. So now they're asking us to sketch the result of a 2D gel electrophoresis for these 4 proteins A, B, C, D. Okay, disregard this part. We don't need to think about this too much. We will just think about what is the pi and what is the molecular weight. Okay, we already know that in 2D gel electrophoresis we will have increasing pi, okay, on this horizontal axis, and we will have decreasing molecular weight on the vertical axis. Okay, so let us just try and think what will happen after the isoelectric focusing, where we are separating based on pi. So here let us say the pi is going from 4 to 9. Okay, that is the gradient that we have established. Then where are our proteins going to come? A has pi of 8.1. So A will be somewhere here. Okay. B has pi of 5.69. So let us say that B is somewhere here. C is 7.7. So obviously it has to come somewhere in the middle. So let us say C is here. And D is 6.29. So just approximately we'll say that after isoelectric focusing, our samples A, B, C, D have come in this order. And now we are loading it into the SDS space gel to run the second dimension. So now they will be separated based on their molecular weight. Again, let us just roughly say that again, high molecular weight is going to be at the top, low molecular weight is going to be at the bottom. So let us just roughly say that we have some marker here that looks like this, 250 to say 10, 500, 50, 10. Okay, let's start with A. It has molecular weight, let's say 62. Okay, this molecular weight is given in Dalton. So this is 62,457. So we'll just say 62 kilodalton. Okay, so 62 kilodalton means now when it travels down, it will come somewhere here. So this will be the final position of A. Same way for B, they're saying 115 kilodalton. Okay, so B will come down to 115. So this will be the position of B. C has a very small molecular weight, it's only 17 kilodalton. So C will be somewhere here. And D has a similar molecular weight of A, it's almost 70. So it will be close to A, but not exactly in the same line. So that will be the final position of D. So this is how the proteins A, B, C, D will separate in a 2D gel. Okay. I think after we've gone through the same concept so many times, this is a fairly straightforward problem for us to solve. Okay. So yeah, I hope this concept of 2D gel electrophoresis is clear to everyone. If it is, I'll just quickly go over some other concepts which Sir covered in his lecture this week. One of them was how to stain different specific proteins. So you already know how we can use like Kumasi stain, silver stain and so on. And those will stain all the proteins in our sample. But we can also stain specifically for some post-translational modifications. For example, if we have some glycoprotein, okay, there's some carbohydrate which is attached to the protein. We can actually detect that specifically using this stain called periodic acid Schiff-based staining. Okay, so here if you see these two gels, we have a marker in the middle and we have these samples. So this is stained by Kumasi and you can see how many proteins are present in this sample. Okay. So this sample 1 is something called horseradish peroxidase, which you already know has a glycoprotein. This is BSA, we already know it is not a glycoprotein. Okay, so this is positive control, this is negative control. Now what we're going to do is do something called this periodic shift-based staining. In that, what happens is, if you have these two OH groups next to each other, the periodic acid will convert it into two aldehyde groups, which is where the shifts-based reagent will attach and form a dye. Right? And this is what we see as a sort of purple, violet, pinkish color in the gel. So if we look at this stain gel, periodic acid shift-based staining, we can see that sample 1, we are getting the same band, right? Because we already know it has a glycoprotein, but we are also getting a band for sample 4. So we're able to see that this band is corresponding to a glycoprotein, right? So this is a way in which you can use specific staining to look at your proteins. Then we can also stain for other things like phosphorylated proteins. So we get commercial dyes. So this is a commercial dye called ProQ diamond. And here it is showing this is a total protein. So here they've used something called Cypro Ruby, which is a fluorescent stain which is going to stain all of the proteins in the sample. So you can see how many sample proteins are there in the sample. But when we stain it only for the phosphoproteins, we see specifically that only these bands are showing up, right? Which is showing these are all the phosphorylated proteins in our sample. And this can be very useful if you're looking at things like signaling cascades. So you have a cascade where many proteins are getting phosphorylated one after the other. And you want to know if that cascade is getting activated. So I want to know if MAPK is becoming phosphorylated MAPK or something like that. So there I can use something like this. Of course, the traditional way to detect phosphorylation is using a Western blot, which I'm sure we'll discuss later in the course. But this is the more traditional way to detect a phosphorylated protein, where you will do a Western blot using an antibody against the phosphorylated part. So here they're looking at some protein called TRPC6, and they're adding protein kinase A. Right? Protein kinase A is a kinase, which means it's going to phosphorylate this protein. So as we increase the pKa concentration, the amount of phosphorylated protein is also increasing. Right? And because we're running this control, we can see that the total amount of the protein is the same. It's just that the phosphorylation is increasing. Okay. So this is how we can look at specific modifications made to our proteins through staining protocols. Okay. Yeah. So again, if you have any doubts, please feel free to ask them in the comments or ask me directly, because this is what I wanted to talk about for SDS or protein electrophoresis. Sir has also discussed some concepts related to nucleic acid electrophoresis, which I wanted to discuss a little bit with you guys. So one was something called DNA autoradiography. So we just discussed ways in which we can detect proteins. And this is another method which is commonly used in nucleic acids for how to selectively detect your nucleic acids. Okay. So you must be familiar that when we run an agarose gel, we'll use ethidium bromide. We'll put ethidium bromide in the gel. That will intercalate with our DNA and give fluorescence. And so that is going to detect all of the DNA in our gel. But this is another method which is more sensitive, and we can use it to label our specific DNA itself, in which we're going to use this radioactive isotope 32 phosphorus. Right? We already know that DNA has a phosphate backbone. So if in that phosphate backbone we have some radioactive molecules, we can use that to detect our strand of DNA. So what you have to do, so what you have to do is basically label the nucleic acid with this radioactive isotope and then carry out gel electrophoresis. When you carry out the gel electrophoresis, you are not going to be able to see anything with your naked eye. So what you have to do is expose this gel to something called an X-ray film. This X-ray film is basically an emulsion of something called silver halide crystals. So what is going to happen is that when our radioactive material hits the silver halide crystal in the X-ray film, it is going to activate the silver halide, and that becomes very reactive. So it is immediately going to become reduced and get deposited as a metallic silver band. Okay. So you are immediately going to see a black band wherever your radioactive nucleotide is present. Okay. So when we develop this film, we get to see an exact image of all the bands that were present on the gel. It's just that we see this black bands wherever the silver is deposited. Okay. And this is what is called DNA autoradiography. So you might have seen gels like this before. This is an autoradiograph where these are the kind of gels that were used originally for sequencing purposes. So this is a Sanger sequencing gel. And basically each band here is corresponding to one nucleotide difference between all these products. And if you want to read what is the sequence of these, then you can have to read from bottom to top to see what is the sequence of these gels. Okay. If you want to quantify an autoradiogram, you can do something called densitometry, where basically you're quantifying how dark is each band. So I can say visually that this is dark, this is light, there is some band here, again there's a gap, one more band, there's a darker band over here, and so on. But if you want to quantify that, that is called densitometry. So I can say that this dark region has some intensity, okay, and that is giving this peak. Again, this region here does not have any color, so it is not giving a peak. And I'm getting this peak again for this dark band. Okay, so this is just an easy way to see what is the profile of our autoradiogram. Okay, it's called densitometric analysis. And using agarose gels, of course, we can look at the size of DNA, size of RNA, what is the nature of these nucleic acids, but we can also look at DNA protein interactions. And that is using an assay called, so the easiest way to do this is something called EMSA, or electrophoretic mobility shift assay. It's also called gel shift assay. This is just a very quick and easy way to see if we are having any interaction between a protein and a nucleic acid. So first what we have to do is mix the protein and nucleic acid mixture, and we just have to run it on the gel. For this, it is important that the nucleic acid has some label. So the most sensitive will be the autoradiography we just discussed. So for example, let us say that we have some 32P labeled nucleic acid. We are mixing that with the protein, and then we're running it on the gel. You can imagine if I have some protein which is going to bind to the nucleic acid, now its size is increased. And that means that it will travel slower than the free nucleic acid itself. So the band will shift upwards, right? And that is called mobility shift assay. There is also one more concept here called super shift assay. In that, so basically here, if I have some mixture of proteins, I'm using it to do this assay, and I'm seeing some shift. That is not telling me which protein exactly is going to bind. In the super shift assay, we add one more component, which is an antibody. Okay? Say we have some idea. Oh, I think this protein might be binding. Then I will add an antibody against that specific protein. If in that case, the shift is more, then that confirms that that protein itself has bound, right? Because, so I have my free nucleic acid, okay? That is going to give me a band over here. I have free nucleic acid with my protein. This is a bigger complex, so the band is going to shift upwards a bit. But say I have my protein, I have nucleic acid, I have my nucleic acid, I have my protein, and I have an antibody against that protein. This complex has become even bigger. So this is going to shift even more, right? So this is called a super shift assay. It is just used to confirm that which protein is binding actually, if you don't already have an idea. Okay. So if you're thinking what kind of gel will I use to run in this, we actually usually use an acrylamide gel. So you will see acrylamide, bis, ammonium persulfate, TMED. all of this is the same as the regular SDS gel that you would use, except there is no SDS, right? It's a regular acrylamide gel, but the running buffer is a TAE buffer. Okay, you have to run it in a nucleotide buffer, but we'll cast an acrylamide gel because we want to basically look at the difference in proteins. Okay, and this is what an EMSA gel will look like. Here we have increasing protein. Right? So as we increase the protein concentration, we get to see the binding of the protein to our nucleic acid, and the band has shifted from here to here. Okay, so this is a very obvious and direct visual measurement of the binding of the protein with a nucleic acid. Obviously, it's not going to be 100, it's never going to be like 100% bound. So you will still get this free nucleic acid band. But you can see by the time we have reached this concentration, the majority of the nucleic acid is bound with the protein. Right? So here they've said they have the cap protein, and they're looking at the binding of this cap protein with the lac promoter. Right? I'm sure everyone here is familiar with the lac operon. So they're looking at the binding of this to the lac promoter. Okay. We can also do some basic stoichiometric analysis here. So now again, here we have increasing protein concentration. And as we start increasing the protein concentration, we actually start getting different bands. It's not like this is just shifting up here and getting one band, right? I'm getting multiple bands, which maybe suggests that there are multiple binding sites for this protein. Sorry. Here they're showing titration of lac promoter DNA with E. coli lactose repressor. Right? And here they are saying that as we increase the protein concentration, what is actually happening is we are getting non-specific binding. Right? So it is binding to multiple sites. So here is the actual binding site, but it's also binding here and here and here. And as a result, the band keeps shifting upward as we start adding more and more protein. Okay. Now, EMSA is a very quick and easy method, but it may not answer all of our questions for this DNA nucleic acid interaction. Now, the first problem, which might be obvious to you already, is that if the protein complex is not stable, right? If it's not stable, then it might dissociate during electrophoresis. And then we won't be able to detect any shift in the band. The second thing is that just because it shifts upward by some amount, that does not mean that shift is directly proportional to the size of your bound protein. Okay, it can also depend on the shape of your bound protein, how it is binding, and so on. So it is not telling you exactly which protein is binding to your nucleic acid. If you want to confirm which protein is binding, then you have to do things like supershift assay, or you can cut the band and do mass spectrometry and so on. So then you get an identity of the protein. That is not identity you're not going to get from EMSA. And it does not tell you which is the binding site in the DNA. It tells you the protein is binding, very good. But it is not telling you where it is binding. Right? If you want to find out where it is binding in the DNA, we have to do something else called DNA footprinting. Okay? What happens in DNA footprinting is the most common way is we can use an enzyme called DNase. What is DNase? DNase 1 is an endonuclease which non-specifically cleaves the DNA. But now imagine I have this nucleic acid, it's being cleaved by DNase. But I have the same nucleic acid and some protein is binding to it here. Okay? It will get cut here, it will get cut here, it will get cut here. But the protein is here, so it cannot cut here. Right? So you can imagine that if I run both of these mixtures on a gel, I'm going to get a very different profile. Right? this is going to give me some set of fragments, but here one fragment is going to be missing, right? I'll get something like this, and one big fragment that wasn't here, because I'm not able to cut it in this middle portion, right? The nucleic acid is protected due to the binding of a protein, and this changes the cleavage products that we are going to obtain. How can we detect this? We can add a radioactive label, say over here. Okay, and now I'm just going to run all of these in the gel. So I'm going to get this set basically of cleavage products, right? And some combination of these. I'm going to run all of them in the gel, and I'm going to see which of these is missing, right? Again, because it's very sensitive, we can use autoradiography. And what we have to do is compare the protected region with the sequencing results, and so then we can determine the binding site. So this is just an example of what DNA's cleavage looks like. So here I have some sample with increasing amounts of DNase 1, and you can see it has formed a lot of bands within this region. Okay, this is the known sequence, and as we discussed earlier, we can kind of quantify the color, or what is the darkness of the band, and we can plot a graph like this. Okay, so here you can see in A, where I have incubated it for less time, all of the bands are up here, so not much cleavage has taken place. So same way, most of the density is here. And as I start increasing incubation time, we start getting smaller products, right? So the intensity starts moving like this. Okay, now let's look at what does DNA footprinting look like. So here it is showing the binding of a repressor to the lac operator. Okay, very straightforward example. So let us look at this. So this lane is the DNA which is digested without a repressor, right? So this is the kind of binding pattern we would expect if no repressor is added. When we're adding the repressor, you can see suddenly this region of the binding pattern has disappeared. Why? Because the protein has bound to the DNA in this region and it's not able to cleave the DNA. Okay, so this is giving us a very direct visual example of where in the DNA this protein is binding. Okay, and if we compare it with sequencing gel, so this is the sequencing gel for the same sample, we can also get the sequence of this region. Okay, so here they have the sequence of this region where the lac repressor is binding. Okay, this is the same thing. Again, it's showing that this is the DNA footprint of the lac repressor. It's not binding in this region. Again, this is showing if I have two proteins, I have a repressor and an RNA polymerase, then both of these can bind to this DNA region, and it is just the repressor. I'm getting this footprint, which means this much region is bound. But once I add the RNA polymerase, this much region is bound. Okay, this has a bigger footprint. So again, this is shown in this graph here. In the DNA plus repressor, the intensity is missing from this region. So this is where the repressor is binding. And if I add the RNA polymerase, then I get an extra footprint here. Right? So intensity is now also missing from here, which is telling us that this is the extra binding site for RNA polymerase. And now, since we can also find the sequence of this region, we can show within the DNA sequence what is the binding site for these proteins. Right? Say I have some transcription factor, and I want to know what is its recognition motif. This is the most easy, most practical way for me to find out what is the recognition motif for my transcription factor. I have to take the promoter for the gene. I have to basically incubate it with my transcription factor, and then I have to perform this DNA footprinting assay. And then I will get to know which region of this is not being cleaved by my endonuclease, and that is showing where my transcription factor is binding. Okay? So what these people were able to show, right, this is a very nice visual representation, I think. You can see clearly that the RNA polymerase has to bind like this over here in the DNA. So this dark region is the binding site that was determined by the DNA footprinting assay. Okay, so this is where the RNA polymerase is binding, this large binding site, and this small binding site is the binding site of the repressor. Okay, yeah. So that was going through the techniques that were mainly discussed in this week's course content. If you have any questions, feel free to ask. Otherwise, I can just quickly go through the problems that sir discussed, and we can discuss how to solve them. Okay. So this is the first problem. It is very straightforward. We have an old rock sample with a dinosaur fossil in the sample. Okay. Now, obviously, we don't know the sequence of the DNA in the dinosaur fossil. So what they've done is they've taken random primers and then PCR amplification. And so now they want to determine the size. And obviously, if I want to determine the size, I should run it on a gel. Right? I will get some bands which will allow me to determine the size of this amplified DNA. Right? So this is a very straightforward problem. Another straightforward problem. PhD student has isolated mRNA from cancer sample. He wants to use this mRNA for northern blotting. But before we go for northern blotting, it's a very tedious technique. First, we should know that the mRNA that we have is actually of good quality. So how should we check the quality of mRNA? Again, we should run it on a gel. RNA gels, as I'm sure most of you will know, they are usually denaturing gels. Because RNA can form complex secondary structures. It can form loops, right? It can form loops like this. It can form what is called a pseudoknot and so on. And so we need to denature all of this and make it linear. Okay? Same way that we denature proteins. So you have to make it linear. So usually we will run it in a formamide gel. But same thing, once we have a denaturing gel, we can run our RNA sample and get bands to tell us what the quality of the mRNA is like. Okay, I thought these two were very straightforward problems. Now we'll come to another one. Scientists have discovered a transcription factor in skin cells which binds to gene X. Now they want to identify the region of the genome where the RNA polymerase binds to transcribe gene X. Right? So this is what we just described. That how do we find the binding site of any protein on the DNA? We have to do DNA footprinting. So what we'll have to do is, this is our gene. Okay. This is our upstream region. Okay, this is gene X. This is some upstream region. So what we'll have to do is take this upstream region, add transcription factor, and then do DNA's digestion. Yeah. How RNA gel is different from conventional agarose gel. So normally in RNA gel, we will add something. RNA is unstable. So that's why you have to be very careful when working with it. So normally in RNA gel, we will add something called formamide or sometimes urea. And how that works is, it's basically a denaturing agent. So it will stop any like intramolecular bonding in the RNA and make it linear. That is the only difference. Sometimes they will also use another buffer called TBE, a trisborate EDTA, which is said to be better for separating RNA. It's said to be better for dissolving small molecules. But those are the only two differences. Yeah. So here, so as I was describing, here we have to perform DNA footprinting. We have to take this upstream region, add the transcription factor, and then digest it with DNase 1, and then run it on a gel. right? So without the transcription factor, we will get some set of bands. And then when we add the transcription factor, we will see that some region of these bands are missing. And obviously this is the region where our transcription factor is binding to the DNA. That's why it is not able to be digested, and that is why we are missing the bands from this region. So again, if we run the sequencing gel on this side, we can know the sequence of this region, and so that will tell us what is the binding site. What is the exact binding site on gene X where this transcription factor is binding. Okay, another problem. We have a MTB strain that is treated with a drug. And because of that, there is a new protein X inside the bacterial cells. Now the patient student wants to determine the molecular weight of the protein from MTB. Again, we just want to determine the molecular weight, so we should run it on a gel, right? Blah, blah, blah. We'll get lots of bands. Only thing is, we want to know what is specifically the weight of protein X, right? So for that, we should have one sample without the drug, one sample with the drug, right? Because we know that the protein X is only coming after we add the drug. And we need to see that if we're getting any extra band over here, which is more intense. And that is corresponding to protein X. Okay. And then if we run a molecular weight marker alongside it, we can use that to determine the molecular weight of protein X. But it is very important to run this control sample so that we can identify that which band over here is actually protein X. Okay, it shouldn't be that we determine the weight of some random band, right? Because we are running all the proteins, we should also run this control along with our actual sample to find protein X. So here, again, here.
Ma'am, I have it down.
Yeah.
Ma'am,
Yeah, go ahead.
the first step, what we should do to determine the molecular weight as while isolating the protein, like first native page or first SDS page.
Okay, so I will assume, let us start from scratch. Okay, I have some new gene. I want to express it. I want to see how it is expressing and what is the molecular weight. So the easiest way to do this is I will just set some small culture. I will take the cell pellet. I will run the cell pellet directly on my SDS page gel. Okay.
Yes, ma'am.
So that is the easiest way, to just start with SDS page. That way you will get all the proteins will separate better. Okay, and you will get to know kind of the true molecular weight of the protein. In the native page, because of folding and it can adopt different, so if I have a globular protein and if I have a linear protein, they are not going to migrate at the same rate in a native page. But in SDS page, both of these will become linear proteins. So they will migrate at the same rate and I will get to know the true molecular weight. Okay, native page will not give you the true molecular weight of the protein. So I would say start with SDS.
Thank you.
Yeah. So yeah. So here we have some protein which can have many oligomers. There is some monomer, some dimer, and some tetramer. The scientist wants to study what is the stability of this protein. So one way to study stability is to use a urea gradient, as is already shown here. So as we increase the amount of urea, we see that the protein starts to unfold. And obviously as the protein starts unfolding, it will also lose this whatever oligomeric status. Okay, and we will get to see the monomer accumulating. So as sir discussed, what we can do is prepare samples in different concentrations of urea. Okay, say I have 0 molar urea, then I have something which is 0.5, 1, 2, 3, 4, so on, up to some 8 molar urea. I'm just going to take the protein and add different amounts of urea, Okay, I'm going to make this urea gradient. And then I'm just going to load it in the gel. So this is my sample with 0 urea. This is some sample with 8 molar urea. Now in 0 molar urea, I am not expecting the protein to denature at all. Okay, I'll run this on native page because I don't want to like have any effect of STS. We'll run it on native page. All the denaturation will be because of the urea. So now in the 0 molar urea, it is going to stay in its original state. So say it is original state is in a tetramer. So I will get a tetramer band. And as we start increasing the urea concentration, we will start seeing denaturation. So slowly I'll start seeing the appearance of the other bands also. Okay. So slowly the monomer band will start appearing. And so we will see what is the concentration of urea required to break all of the oligomeric states. So say this is 3 molar urea. And at 3 molar urea, I am not seeing the oligomeric state anymore. So that is giving me an idea of the stability of the protein. So I can do this with multiple proteins. And I can study what is the amount of urea required for denaturation. And so that will give me an idea of like which protein is more stable and which protein is less stable. Okay? Just, yeah. Maybe one last problem we'll do. A scientist has isolated a unique protein A, which is responsible for induction of apoptosis. He suspects that the protein A is interacting with the DNA polymerase and disrupting its replication. Okay, he just wants a quick and easy experiment to study whether protein A is actually interacting with DNA polymerase. Okay, here it is asking to look at protein binding. Okay, there are n number of different experiments you can do to show protein binding. Okay, so I'm just writing the names of some techniques here. These are all techniques we can use to check for protein binding. Okay, but all of these are very advanced techniques that will require a lot of protein and they're not very easy to do. So the quickest and the easiest way will be just to purify protein A. So I'll call this protein A. We'll call DNA polymerase protein B and just run it in the gel. Okay, so here I have protein A, protein B, protein A plus B. Run it in a native page. I'll get some band for A, I'll get some band for B. If in the mixture of A plus B, I'm getting some other band up here, which is corresponding to the complex, then that means that this A plus B complex actually exists. Okay, that means that these two can interact with each other. Right? So this is by far the easiest way to look for protein binding. There are many other ways in which you can do this, but this is by far the easiest. Yeah. This is another method where, again, you can have protein A and basically interact with protein B and then do a Western against protein B. Right? So again, if you're getting signal here, that means that A and B had to bind in the first place. Right? So like there are many approaches to do this, but this is the easiest. And yeah. Okay, we'll discuss this problem. I think it's more interesting. We have discovered a transcription factor that binds to gene X. Okay. Now they want to find what is the identity of this transcription factor. Right? So how can we find this? So you may have heard of techniques like CHIP. Wait. You may have heard of techniques like CHIP or something like that, where you can identify what protein is present on the gel. But another easier way is you can just do EMSA. Okay? So we can have this DNA region. This is going to be the nucleic acid, the labeled nucleic acid. and we can put our cell lysate along with the nucleic acid. And if we are seeing a shift in the gel, that means something in our cell lysate has moved, has bound to our protein, has caused a shift in the EMSA. And again, earlier we discussed the concept of an EMSA super shift assay, in which you also add an antibody. So if we add an antibody against our suspected transcription factor, and we're getting a bigger shift for that, that means that then we have identified that which transcription factor is actually binding to this coding region. Right? So EMSA is a very quick and fast way to just determine which, if a transcription factor is binding, and then we can use other methods to confirm which is the actual identity of that transcription factor. Okay, so yeah, that is as much as I wanted to discuss today. We just went through the techniques that Sir had discussed in the content, and also went through the problems that he had discussed. If you guys have any questions, I'm happy to take them. We can discuss if you have any doubts in what we discussed today or any of the course content.
Actually, I have a question, but not from today's class.
That's
that
okay.
is, we are going to have exam, right? At the end.
At the end, yeah.
Yes. So is it like driven towards only Sir's course lecture, or from the PMRF lectures we are also getting? So
No, it'll only be from the Sir's coursework.
okay.
This is just additional information.
And I don't know, is it with me or with everybody? I am not able to access the slides from NPTEL. I mean, it is showing like you don't have access, request access. So yeah.
Okay, so this is this is for the live session, is it? For the, if you go to the
Yes, yes,
Google Drive, you're not able to access.
yes. The material, I am able to see the video link of the lectures, but I am unable to access the material.
Okay.
Whole material.
Okay, so I'll just check, because the material is uploaded from my side, so I'll just make sure that it's available publicly. Yeah. Thanks for letting me know.
Yes, actually, it is uploaded by everybody, but I am not able to access any one of them. Is always showing request access or something kind of that.
Okay. Have you tried to request access?
Yes, but I think I didn't get any response.
Because I didn't get any notification also that someone is requesting access. Okay, I'll just make sure that it's available from my side, and maybe I'll.
I don't know how these windows work. I mean, is it through mails or something? They get notified that yes, someone is trying to get the access, or I don't know.
No, so it's hosted in Google Drive, no? So since it's hosted in Google Drive of my Google account, I should get a mail that someone wants access, but I haven't gotten any mail.
I don't know. Actually, I tried this yesterday and the day before yesterday also, but I didn't receive any answer or reply.
Okay. I'll just check from my side, and I'll send the NPTEL organizers a mail saying that you guys are facing this problem.
Okay, ma'am.
Yeah.
Thank you. I don't know if it is just with me or everybody. I'm just saying from my side only.
Yeah. So,
I don't know about others.
So if anyone else can just check and let me know in the next session if this problem is still there.
Yes, I will check. I have not checked.
Yeah, sure. Just let me know by the next session if the problem is still there. From my end, I'll try to find out what is going on.
Okay, ma'am. It will be very helpful, ma'am. Thank you.
Sure.
I mean, it's not possible to see the 2 hours of lecture.
Obviously, yeah.
So it would be very helpful that I could just give it a glance to the lecture. I mean, the PPTs.
Yeah, of course. That's why we upload the PPTs, no? So.
Yes. Thank you, ma'am. Thank you.
Yeah, sure. No worries. Yeah. If there's anything else we can discuss, or else I'll call this session closed.
No, ma'am. Nothing. Yeah. Thank you, ma'am.
Sure. Okay. Thank you, guys. Okay.
Good night, ma'am.
Good night.